So welcome everyone. This hopefully will be the last online meeting of Cafe Sci. We're going to start again in the club next week, in the new Headingley club. So next month, sorry. So please come along if you can, and enjoy a beer in the bar afterwards, which we can't do today, unfortunately. So our speaker today is Nishant Ravikumar. He's a lecturer in the School of Computing at the University of Leeds. He has a background in machine learning for medical image analysis, and he's part of the Centre for Computational Imaging and Simulation Technologies in Biomedicine. He's going to be talking about computational imaging and simulation technologies in biomedicine, funnily enough. So thanks very much for coming, Nish, and over to you.
Thank you very much, Dave. It's a pleasure to be here. Thank you for having me. I'm just going to share my slides and we can get started. Okay, can everyone see my slides?
Yeah, that's fine. Yeah, I can see those. Yeah.
Excellent. All right. So let's start with just a brief background about myself. So I'm originally from a city called Bangalore in India, which is in South India. It's a relatively small city with just 10 million people. By Indian standards, that's just 4th on the list, so not the biggest, amazingly. So I moved to the UK for my undergrad in 2008, where I joined the University of Sheffield. During my undergrad, I spent a year in the US as part of my studies at a university called Case Western Reserve in Cleveland. Following this, I did my PhD in machine learning for medical imaging, which is what I'm going to be discussing with you today. At Sheffield as well, between 2013 and 2017. And then I spent a couple of years as a postdoc in Germany, in a city called Erlangen, which is near Nuremberg, a nice, quaint little town where I learned a lot about my field. And this gave me the opportunity to rejoin SysTib in 2019. And I was recently appointed as a lecturer in the School of Computing at the University of Leeds. So just to give you an overview of the work we do at SysTib, you could say that our work focuses on two main areas of research. One area that looks at developing machine learning algorithms to extract information from different types of medical data to help with identifying patients at risk of future disease, improving our understanding of how disease might progress in specific patients, and improving our ability to understand how disease mechanisms vary between patients and what effective treatment strategies are. So on the one hand, while we focus quite broadly on how machine learning and other tools can be used to analyze medical data, a large portion of our work is focused on cardiac imaging data, or not just cardiac imaging data, but cardiac images and data related to assessing cardiac health. And a portion of, or a part of today's talk will focus on a recent study that we participated in, where we looked at whether we could identify patients at risk of future heart attacks based on learning information from cardiac images and retinal images, believe it or not. But we'll get to this in a little bit more detail later on. The other area of research that we focus on is this new field of in silico trials for medical devices. So in silico trials, as the term suggests, means computer generated or virtually orchestrated trials where you can examine the performance of a specific medical device and how it might interact in a virtual or digital setting. And the power of such a paradigm is in that it allows you to explore the performance of medical devices in a much wider range of parameters than what you could possibly do within conventional clinical trials that are used to assess medical device safety and efficacy prior to their deployment in markets. So I'm going to give you a flavor for the first aspect of our research, where we look at predicting future chance of disease or future heart attacks in patients, and then also give you a flavor for how this paradigm of in silico trials and modeling interventions virtually can help accelerate the development and help improve the safety of medical devices. So the question that we wanted to answer is, can we say something about the heart without looking at the heart? And you might ask, well, why would you want to do this? So let's take a look at why this is a problem that is worth approaching, or why this is interesting. So cardiovascular diseases are the leading cause of mortality and morbidity worldwide, with the World Health Organization recently reporting that an estimated 17.9 million people per year die of some cardiac-related disease. The detection and diagnosis of cardiovascular diseases and identifying patients at risk very often rely on cardiac magnetic resonance images. And the animation that you see in your bottom right is what is known as a cinematic cardiac magnetic resonance image. So what this shows you is the contraction and expansion of a patient's heart across a cardiac cycle playing on a loop. So cardiac magnetic resonance images contain a lot of information that can be extracted about the structure of the heart and whether the function is normal or abnormal, or whether there is some pathology that affects the normal functioning of the heart. The way these images are analyzed at the moment are through manual assessment, so by expert cardiologists who have seen millions of these images and can just look at a cinematic sequence such as this and say, well, this person's heart has an enlarged myocardium, which is the wall of the heart, and hence prescribe specific forms of treatment or ask them to undergo additional tests. There are also semi-automatic approaches which use some computer algorithm along with some user interaction to extract specific types of measurements that might be instructive about whether the heart structure and function is within what is expected in a normal population or whether it is abnormal. And we're slowly starting to see an increase in the number of fully automatic approaches, fully computer algorithm-driven approaches that can satisfy some aspects of this assessment. And we're slowly starting to see these being adopted in a clinical setting. But the fact remains that MRI systems are extremely expensive, not just in procuring one, but in their maintenance. And being able to interpret MRI images requires specialized training, which again can be is a very expensive endeavor. So this means that MRI-based analysis of cardiac structure and function, while being extremely useful, is not easily accessible for large proportions of the population in the world, especially in developing countries like India, where you have a very large population, many of whom are at risk of cardiovascular disease, and there is limited infrastructure for them to get access to such facilities, or for indeed such facilities to be able to deal with the size of the population there is. So what can we do to address scenarios like this, where relying on cardiac MRIs is insufficient to identify patients at risk of cardiovascular disease. This was what we wanted to answer in this first body of work that I'm going to speak to you about. So the natural thing to think is, well, okay, if we can't look at the heart because the types of images and the imaging systems are inaccessible or very expensive, then can we look somewhere else in the body using some other form of measurement that or what potential risk you might be at in the future. So in the last few years, there's been a number of different studies that have looked at whether the vessels in your eye are somehow instructive for understanding your overall vascular and cardiac health. So one of the seminal studies in this domain was this paper published by Google Health in the US a few years ago, that for the first time demonstrated that using these really cost-effective retinal photographs, which you see in the top right here, that they were able to predict certain cardiovascular risk factors. So the idea here is that these fundus photographs are quite high resolution depictions of your retina, along with some of the blood vessels that you can see there. And these blood vessels, depending on their tortuosity, the number of bifurcations, whether they have occlusions, etc., encode some information about your overall vascular health. So what this study explored and showed is that by learning certain hidden representations from these retinal images, they were able to actually, for the first time, not just associate, but predict cardiovascular risk in patients. So we wanted to take this one step further, and this definitely formed an inspiration for us. And we wanted to see whether, instead of just predicting cardiovascular risk factors, we could actually predict risk of future adverse events, such as a heart attack. And that brought us to this study that we published just earlier this year, about a couple of months ago. And the key difference in what we tried to do relative to what has been done before was to see whether we can learn some common representation, not just using retinal images and patient data, but combining patient metadata such as their age, gender, their blood pressure, cholesterol, all of your standard clinical measurements with retinal images and their corresponding cardiac MRIs. So you might think, well, the whole purpose of doing this was to avoid having to look at their cardiac MRIs, because the entire motivation is to have a cost-effective way of analyzing their risk of cardiovascular disease. So the key here was, can we build a model that, when being learned, utilizes all of these sources of information to learn some hidden representation of about how susceptible they are to disease, such that when it's actually applied in a complete test setting, it only requires the patient's retinal images and some minimal personal information, such as their age and gender. So again, the motivation here is that if you went to an optician or an ophthalmologist, you're unlikely to have a lot of clinical measurements taken. At the most, it would be your age and gender that's recorded, along with this retinal photograph that's acquired. So if we were able to use just this information to say something about your risk of future disease, or in this specific case of myocardial infarction, then that would be a very powerful tool, because you could expand this to really wide and large populations in countries or settings that have limited access to advanced imaging centers and limited access to cardiologists and experts that are required to interpret data acquired in such advanced imaging systems. But the question is, where can you find such a nice data set of cardiac MR images, patient metadata, and their retinal images for all the same subjects and at scale for a large number in a population? Luckily, the UK is one of the few places in the world where such a data set is available. So the UK Biobank is a large-scale biomedical data collection initiative that's been running since, I think, 2006 or 7, if I'm not mistaken, and has collected biomedical data for about 500,000 participants in the UK. So this formed the perfect testbed for us to develop our idea, test our hypothesis, and to see whether we can, in fact, predict future heart attacks in people based on just their retinal scans and some demographic information. And this is exactly what set us on this path. I should say that before we got to analyzing this combined data, there was a lot of legwork that went into first just analyzing the cardiac MRIs alone. Because the first thing you need to do if you want to extract any kind of quantitative information from a cardiac MRI automatically and at scale across 40,000 subjects in this scenario is you need a way to separate the tissue that belongs to the heart from the surrounding tissues in your body. Once you can do that, you're able to compute specific volumetric indices and other types of quantitative information that tell you something about how your heart is functioning. So what you see on the left here are the results of our automatic algorithm that's able to separate the heart from the rest of the tissue, where the blue contours are essentially showing you that what the algorithm identified as belonging to the heart and everything around it as belonging to the background. So this was necessary to even enable training such a system, because we only wanted to look at the heart and prevent, surrounding tissue information from influencing what is learned together with the retinal images and demographic data. And just to show you something that is quite unique, this was the first study that ever computed the variability in the volumes of the heart across such a large population for the full cardiac cycle. So what you see here on the right, the curves and their standard deviations, are representations of how different volumetric indices varied across 40,000 subjects entire cardiac cycle. And this type of analysis has only become available in recent times due to technological advances, both in terms of machine learning algorithms, but also, most importantly, in terms of computational infrastructure. So coming back to our study. So we had this groundwork laid down for us from previous studies that we had participated in. But the question was, how can we learn a shared representation from such multimodal data from cardiac MR and retinal images, together with some non-imaging data like clinical measurements and patient demographics? Well, again, we looked into the most recent advances in machine learning, and we found that there is a way to build a system to learn combined latent or hidden representations from multiple channels of data. With the key idea again here being that once we learn or extract this hidden information, this can be applied to any new patient's data without needing for that data to be complete, right? And what this means is that if we could train it or build it using retinal images and cardiac MR images, then when we wanted to test it on a new population, we could make do with just the retinal images. And while this approach is quite generic and could could be used to assess patients' risk of a variety of different types of diseases, not necessarily specific to cardiac. We focus this study on identifying patients at risk of heart attacks, as we found that there was a large incidence of these within the UK biobank population. So that's all very good, but what if we want to also look at how this fares in different scenarios? So we talked about how specific types of clinical measurements and demographic data are only available in a hospital setting and are not available in, at standard opticians or eye clinic. So part of this study, or a large part of this study, also focused on identifying which of these demographic variables that are listed at the bottom here that are established as standard cardiovascular risk factors, which of these contribute the most to having an accurate prediction, and which we could do without. Because if we could do without most of these, then the more powerful or flexible our overall system is, because it would mean that with minimal information, we'd be able to make valuable predictions about a person's risk of future heart attacks. So what we found through building this system was that actually we can predict future risk of heart attacks pretty well. So what these curves show you is a metric called the area under curve and the ROC curve. I won't get into the details of what they represent exactly, but for now we could very loosely use the term they represent some form of accuracy. So if you have a perfect curve, you would have 100%, your curve would be right to the top left here, and you would have 100% ability to identify patients at risk of future heart attacks. And what these demonstrate is if we actually include just retinal images and metadata once our model has been trained, there is a significant improvement compared to using metadata alone. And this was an extremely important finding for us, because it demonstrated for the first time that by learning combined representations with cardiac MR and retinal image data, you were able to extract whatever hidden information was there in both to subsequently identify patients at risk of heart attacks using just their retinal images and associated patient information. So that's great. But, as we said, blood cholesterol and glucose and all of these other measurements may not be available. So how does the performance fall or get affected if we have less demographic information? And as I said, the main motivation here is to see how we can make this process of identifying patients at risk the most cost-effective possible. So if it reduces the need for conducting blood tests and for them to visit hospitals to get additional clinical measurements done, then it's going to reduce the overall cost of delivering patient care. So in the scenario of an optician or an eye clinic, we could say that the only reasonable things to expect to be collected in terms of patient information are their age and gender. And again, what we found is that the performance of our model during test time, once it's been built, wasn't significantly affected when we reduced the amount of additional patient metadata that was available. And again, this was an excellent result for us because it helped us test and demonstrate the hypothesis that when you extract the shared representation with a rich set of features or types of data, you're able to extract information that you may not be able to find by considering each data source individually. And through this, we were able to also show that there is a potential here for deploying such a system in settings that are looking for a cost-effective solution for identifying patients at risk of future myocardial infarction. So to summarize just this portion of the talk, the points that I'd like to emphasize and for you to take away are that retinal imaging really does show promise for identifying patients not just at risk of future heart attacks, but of developing cardiovascular diseases. And this is something that we are currently exploring with students in our lab. Another interesting question that pops up is, well, if you can use the eye, then are there other organs that you could look at that also provide some complementary information that can tell you something about your heart? And this is certainly the case and is something that we are pursuing that we're going to pursue in the near future. And finally, expanding to larger populations requires that this is a cost-effective way of assessing patients at risk, which is what this study demonstrates in a controlled setting. But to be able to assess larger populations and to test this in a real-world setting, we would need to first conduct extensive clinical trials to assess the safety and efficacy of such computer algorithm, and hopefully through those clinical trials get regulatory approval to actually deploy such a system and have a positive impact on improving patient quality of life. So that brings us to the second part of my talk, which is slightly tangential, but not completely unrelated. So it's this idea of conducting in silico trials to evaluate medical devices and how medical devices might interact with patient anatomy and physiology. So what are in silico trials? Well, we get to that in the next couple of slides. The focus for our work in silico trials is on what are known as TAVI devices. So TAVI devices, which is a short form for transcatheter aortic valve implantation devices, are used to treat aortic valve stenosis. So when the aortic valve gets stenosed, it restricts the amount of blood that's able to flow out of the heart, and this causes all kinds of problems. So a TAVI device is placed to open up the valve and to act as an artificial or surrogate valve to ensure that there is normal blood flow and distribution across your body. TAVI has found quite an increased amount of use in recent years because in terms of patient risk, it's less invasive than, let's say, a full surgical aortic valve repair. Certain studies have demonstrated that short-term hospitalization following treatment with TAVI devices is lower compared to full surgical repair. And in general, there's quite a lot of candidates who unfortunately need to undergo TAVI operations annually. So this is around the figure of about 180,000 patients across the EU, US, and Canada. And these patients are usually, or so far have been deemed high-risk candidates that are in need of TAVI operations. So what high risk means is generally people who are beyond a certain age and have a certain degree of stenosis in their aortic valve, such that surgical operation is considered too high a risk, and hence TAVI is the only alternative. But in the last couple of years or so, with the improvement of TAVI devices, various regulatory bodies have expanded their use to low risk and younger patients as well. So this is all to say that TAVI is quite a promising form of treatment for aortic valve stenosis. There is an increasing proportion of the population that would be that could be treated with TAVI as opposed to, let's say, 10 years ago. But the problem remains that the overall procedure and the act of implanting a TAVI device is extremely expensive. And this is primarily driven by the high valve cost. And this is a common problem that we see across a lot of medical devices that are used to treat various types of diseases. So some of the common issues with the innovation pipeline and the development of medical devices and TAVI devices is that clinical trials, conducting the extensive clinical trials that are necessary to go from conception of an idea of a device to actual development and initial testing to evaluating in animals and first in humans and so on, is quite an elongated and expensive process. The second aspect is that you can imagine that recruiting a wide enough demographic of patients to such clinical trials can be very difficult, and in a way almost intractable to get the kind of variability you would need to know exactly how a device is going to perform in various combinations of anatomy and physiology. All of this impacts the time it takes to get a medical device to market as well, because conducting these clinical trials and being able to recruit patients usually takes years to complete, which means that from the time an idea for a device is conceived to when it actually is available on the market to improve someone's quality of life or population's quality of life, it's usually anywhere between 3 to 6 years, if not longer. And finally, another really difficult thing to deal with is late phase failures in medical devices, so late phase clinical failures, because this is at the point where they've gone through a number of regulatory checks and they are in, let's say, the final phase of clinical trials. And if medical devices fail at this stage, it can have a catastrophic cost to human life. So all of this means that if we had a better way to inform the development of these devices in the first place, inform the types of patients or populations that they are suitable to treat, reduce the number of patients that need to be recruited for clinical trials, it would not just decrease the amount of time to get these devices to market, but it would reduce the cost to human life in the overall process. And as far as the medical device companies are concerned, it would reduce the cost of manufacturing these devices. And this is where in silico trials come in. They provide this tremendous potential to reduce the number of patients that might need to be recruited and the number of phases of clinical trials that might need to be conducted, to refine the types of hypotheses that are being tested in these clinical trials, and to refine the overall process of analyzing or assessing the efficacy of these devices, and ultimately to replace the need for certain aspects of clinical trials that are currently part of the standard operating procedure to get devices to the market. So in silico trials, try to do this one basic thing. When you have a real clinical trial, you are restricted in terms of the patients that you can recruit. You're restricted to analyze just a certain combination of anatomical, physiological, and device design characteristics. And this is because you can only recruit a finite number of patients that fit the inclusion and exclusion criteria that are necessary for testing a specific type of medical device. And this means that when you're evaluating the performance of any medical device, you're looking at this type of grid on the left, which represents the combinations of anatomical, physiological, and device characteristics that have been tested. So the black dots here represent, let's say, a sampling in this clinical trial space, where each dot says that this specific device has been tested in patients that have this range of anatomical and physiological characteristics. But what this also means is that it's almost impossible to get a thorough evaluation of a range of medical devices across the anatomical and physiological characteristics you might find in a general populace. And that, at its core, is where in silico trials show so much promise. Because if you could have reliable models, virtual models, that represent anatomical, physiological, and device design variability, then you're not limited by the number of patients that you can recruit. You can conduct extensive simulations, virtual simulations, that assess device performance in various combinations of anatomy and physiology. So what this would allow is, to reiterate, reduction in the number of late phase failures that medical devices undergo, and reduce the amount of cost that it takes to get a medical device to market. And conducting in silico trials in this way would help exploit the tremendous amount of data that we seem to be collecting on an ever increasing day-to-day basis nowadays, to actually build representative populations of virtual patients, and through these to accelerate the development and innovation of medical devices. So to kind of give you an overview of how we envision the overall process of an in silico trial, what we might start with is, let's say, multiple data sets that don't come necessarily from the same patient or even from the same country, but are complementary sources of data about anatomy and physiology from across the world. If we could combine this information in some coherent way to build virtual patients that describe plausible variations in anatomy, physiology, and pathology, then we could start experimenting with virtual implantation of devices in these virtual patients. And through using established means of simulating biophysical phenomena that look at the interaction of devices such as a TAVI device with anatomy and physiology once implanted, we could make certain inferences about patient outcomes based on the underlying anatomical, physiological, and device characteristics. And what this would allow is, for instance, to look at a single virtual patient and how they respond to treatment with a variety of medical devices, right? So if you consider just a single dot here and this single dot in the bottom right corner of this grid to represent one virtual patient, an in silico trial would actually provide you the ability to look at how this patient responds to a variety of different devices used to treat their particular disease, or in this case, to treat aortic valve stenosis. And this type of analysis, needless to say, is impossible in the real world, because you'd never even try to implant multiple devices in the same patient, let alone ask them to undergo multiple tests with different devices. So this type of paradigm to evaluate virtually the performance of devices in as many combinations of anatomical and physiological characteristics as you can build through multiple sources of data is extremely powerful. And this is the potential that we see in conducting in silico trials for medical devices in general, and for TAVI devices in particular. So just to give you a very quick example of what building a virtual population might look like, you might say, well, okay, you say, you can build a virtual population, but you still need data. And if you need data, it means that you need to get this data from real patients or at least some volunteers, which means you need to recruit. So where exactly are you reducing the amount of cost involved in this overall process? Well, the key is that we have already collected a lot of data across the world. And this data collection process is ongoing and continuous and is increasing for several other purposes. But there is no coherent vision to fuse the amount of information that is contained in all of these disparate data sources to build plausible virtual patient models. And if we can do this, then what is depicted in a very sort of hand-wavy fashion on this slide might be possible, where I can get CTA images from a population of, say, patients in India. I could get cine or cinematic MRI images from some patients in China. And I can find a way to extract the information I need for the anatomical or physiological characteristics of interest and find a way to combine these such that if I sample from this combined model, I'm able to get realistic representations of cardiac anatomy and physiology that are representative of what you would see in a combined population of the Chinese and Indian demographic that I used. And if we could do this at a much larger scale, then you could see that really the sky is the limit as to how much variability we'd be able to capture in the virtual patient populations we generate. So I'd like to end my talk with just a few points on the value and the potential we see for in silico trials. So we truly believe that this paradigm of in silico trials has the potential to revolutionize medical device innovation. We are still very much at an early stage as a group, and the in silico trial community in general, in terms of realizing what is achievable through the technology that comes out of this line of research. They have the power to inform the design of clinical trials, prevent or reduce the cost to human and animal life through that is inherent in conducting clinical trials in the development of medical devices, and to ultimately improve patient safety once medical devices are approved and available on the market. Both of the exemplars, research exemplars that I've covered with you today, I truly feel are results of the technological advances in artificial intelligence and, most importantly, in computational infrastructure that we've seen over the last 10 years. And we are at a crossroads right now where these technological advancements have provided us with a unique opportunity that we've never had before to improve patient care, not just in terms of preventive care, but in terms of the types of treatments that are available to them and the timeliness of those treatments being available to them. And with that, I'd like to end my talk, and I'd just like to say that the research I covered in my talk today would not have been possible without the support of my entire team. So really, this is a reflection of all of the hard work that they've put in over the years, and it's my greatest pleasure to be able to share it with you. And thank you for your time, and I'm happy to take questions.
Thank you very much, Nish. That's really fascinating, really interesting talk, which I found very interesting anyway. about developing all those techniques, various techniques. Can I just remind people that you have to leave in about 15 minutes or so, I think. So if anybody has a question, please indicate, put your virtual hand up. Or John there, I see indicating.
Thank
John,
you,
go ahead.
yes. Good evening, and thank you for an excellent and very interesting talk. May I inquire, it's evident that you chose to look at the vessels in the fundus of the eye simply because they're accessible and uncovered. They're not buried in tissue. That's one thing. The second thing is that there is the presumption that you would find similar, presumably atheromatous changes in the arteries in the fundus as you would in other major arteries like the aorta.
Yeah.
That my experience of looking at these things is that development of atheromatous plaques is in fact very patchy. So presumably you've got a big enough sample of arteries in the fundus to be able to do that usefully.
Yeah, so you raise a very good point, and I think I probably didn't cover this in sufficient detail. But what
you had a lot of things to cover.
I did, yeah. In hindsight, maybe too much. What we were looking in the fundus was not necessarily for changes related to buildup of plaque in the aorta, but was for general vascular changes. So there we were more interested in identifying any type of information that would say predispose a person towards a heart attack. So in terms of ischemic heart disease.
Yep.
So it's more looking at whether there's anything in the vascular structure in the retina that tells you something, I guess, about vascular pathologies in the coronaries.
Yep.
The aortic bits are more related to the TAVI work and weren't part of what we were looking at in the fundus study. But you are absolutely right that we needed a lot of data to be able to do this. In fact, I think the total number of fundus images were about 80,000, belonging to 40,000 participants. And there were a fewer number of cardiac MR images, but the way our model works is that it can leverage missing information, even if you don't have the same two types of information from a single subject. As long as there is sufficient overlap in the data set, it's able to learn a meaningful representation of the features that can enable with the detection of future cardiovascular risk.
It's a sort of superimposition.
Yes.
Type of technique. Yes.
Exactly.
Good. Thank you very much.
Cheers.
Thank you. Paul, you have a question.
Yeah. Well, first, say thanks very much for a very interesting talk. I mean, I had a little bit of association with clinical trials, and to see if you can make them more sort of efficient, and you don't have these failures at right at the very end when there's already a lot of time and money and whatever you expense. So that's all to be good. And I just, presumably when you're doing it, I mean, was checking all the way along that you kind of, because you know things. Well, there's always the possibility of going off on the wrong path, as it were. But actually, the one point I was going to say, a simple point really, was you mentioned about age and gender for your predictors to add in the model that you can easily accept. Surely BMI or something like that would be pretty easy to get as well. You need some scales and
I mean, you're absolutely right. From a purely logistical point of view, I think they would be pretty easy to collect. But we had to defer to an ophthalmologist that was part of our study in deciding on these variables, and to ask them, what types of information would be possible to collect, and what types of information would patients willingly provide when they attend a sort of eye checkup. And the ones that were recommended to us were age and gender, but certainly I could see how with the consent of patients, something like BMI could reliably be collected, even if it didn't mean having to measure someone's weight at an eye clinic.
Oh, okay, yeah. You just, yeah, I suppose it all has to be done with consent. You can't just have somebody just say, well, this person looks a bit overweight to me, but yeah, I mean, it'll be because presumably, I don't know, I mean, I'm very welcome to this because I've suffered with that atrial fibrillation in the past, and maybe I should keep an ear to your research for future need, as it were. But what was I going to go say? Yeah. Oh yeah. So that's enough for me. That's fine. Forgotten what I was going to say there. Okay. Cheers.
Cheers.
I was just wondering, way back in the past, many years ago now, I was involved in modeling stents, arterial stents, just from an engineering point of view to see basically how they worked and how they might be improved. And I was wondering if the research, I think you included in some of your diagrams some of the kind of actual devices that might be used to compensate for some of the problems that people have. Does that also include different designs of different devices, or how far has that come along now?
So it's certainly within our plans to be able to evaluate different designs of stents or devices within virtual patient populations. In fact, that's the entire benefit, as we see it, of doing in silico trials, because you're able to do this type of head-to-head comparison in exactly the same setting that is just not possible in the real world. In terms of stents themselves, I didn't include it in this part of the talk, but we have done actually quite extensive work with stent-like devices that are used to treat cerebral aneurysms. So there are these devices called flow diverters that are used to divert blood flow into the aneurysm to help with occlusion and the formation of clots, and hence the treatment of aneurysms. And something actually we're looking at the moment is comparing different types of devices that can be used to treat intracranial aneurysms.
Right. So yeah. So that's part of Okay. I can see how the modeling would help with that. Absolutely. Yeah. Okay. Thank you. Anyone else with any points, questions?
I mean, so how's it progressing? I'm presuming Leeds is not just the only place that's doing this, what seems to be very important work. So, I mean, presumably it is part of a sort of kind of global revolution. Am I right in thinking that?
Yep, you are absolutely right. In fact, the FDA in the US released a set of guidelines and standards for approving systems that are created to conduct in silico trials not too long ago. So there's an entire regulatory body and documentation that sets out what would be required to conduct in silico trials in the right way, such that they could be used to assess the safety and efficacy of medical devices. And yeah, certainly this is a global revolution, and there is quite a large community within the UK as well that's looking at empowering the use of in silico trials for improving medical device development and innovation.
I suppose just to follow up, I mean, do you know when, what the next steps are? When will we have a device which has been, don't expect you to a crystal ball, but when might this sort of become more?
So I know that there is actually one clinical trial that's being conducted at the moment in the US, I think, where there is a parallel in silico trial that's running alongside to help inform the decisions they make during the clinical trial itself. And I think that's the first of its kind. So I think we are definitely on our way in terms of it becoming part of the regulatory approval pipeline. But I think we are still probably a long way away from getting rid of the need for large proportions of clinical trials.
Thanks a lot. Peter, I think this will be the last final question. You have to unmute yourself though. Can you?
You need to unmute.
Yeah, I think it's.
Yeah, done it. Sorry, I was trying to find the button.
That's it. Yeah.
Thank you very much for a very interesting talk. My question relates to the clinical trials section and how you deal with as yet unknown interactions between individual characteristics, because you're generating a synthetic population and you're using information about the effect of a particular characteristic on the likely interaction with the item. But what if there is an interaction between characteristic A and characteristic B in respect of its or their combined impact with the device? How do you deal with that? Are you confident you can do that?
Well, that's a very interesting question, and let me answer it in two parts. In the first part, when we synthesize these virtual populations, the idea is not to sample from our model completely at random. We would want to have a controlled synthesis of of these virtual patients such that they meet certain criteria as you would have inclusion and exclusion criteria for a real clinical trial. The difference is that we have the flexibility in expanding these inclusion and exclusion criteria without the need for, let's say, regulatory approval or anything like that before doing so. So this would allow us to control what types of characteristics in terms of anatomy and physiology are present in the virtual populations we synthesize. As to whether we know how to account for the complex interactions between these and the device that's finally implanted, at the moment, I don't have an answer for you as to how we could tackle this reasonably, because we just haven't got there as yet.
Yeah. Thank you. Tricky one.
Paul, were you going to say something?
So, I was just going to say, so like, just to say to other people, interactions a bit like you mentioned age and gender. I mean, would you, so that would just be that, the older women would behave differently to older men in that kind of multiplicative sort of way. Yeah.
Well, I was thinking more of a more unlikely combination of characteristics where it just might be that it would be a complicated between age and gender and BMI and, I don't know, ethnic origin or something.
That it's
very
more
high order
of
interactions. I'm just saying that as an example how the effect of age is different for men and women. Say
So I mean, there is one aspect of our work that will need to focus on designing new ways of evaluating virtual populations in themselves. Like how can you say that something is plausible, right? Because what you're describing is you can have these interactions that may not ever co-occur together. And we would need a way to, or we would need to develop a metric that tells us in some objective way whether this is plausible or not. And that is definitely tricky, but it would be necessary to evaluate the quality of any virtual patient population that we synthesize.
Okay, thank you very much. Nish, I think you need to go now. But,
I do, yes. Apologies for having to leave.
No, that's fine. Thank you very much for coming.
Have you? Yeah, thanks. Have you asked about being able to put stuff on our website for the others that haven't been able to make it today?
Yes, we're recording this, and that will go on. The link to that will go on the website, so people can catch up later on, and then inundate you with lots of emails about what you've said. No, they probably. I'm sure they won't. But thanks ever so much.
Well, thank you very much.
Pleasure was all mine. Thank you very much for having me.
And thanks to Amy for putting you up to it, whether you liked it or not.
Thank you, guys.
Okay.
Thanks for giving us a platform to talk to everyone about what we do. Thank
No,
you.
it's really interesting.
Very good. Very
important.
Cheers.
Yeah.
Have a good
So,
evening.
Yeah, you too. Thank you.
Thank you.
And we'll
Bye-bye.
see everybody next time in the club. So
Yeah.
it's in the club.
What is it?
Have a drink this time. Bye-bye.
Bye-bye.
Thank you.
Bye, folks.